Welcome to Chapter 3, Topic 2. This topic covers sections 3.2 and 3.4. It's kind of a sandwich about the structures of the cell as opposed to the cell envelope, which we've pulled out and made into its own topic. This topic is important because we're going to discuss all the structures and their functions within the cell. Without this, we're going to have a hard time understanding how the cell works. So this picture is the sum up of this topic. Basically, if we could, we'd just have a 10-second lecture that says, memorize this picture. This is a picture I want you to know. I want you to know what all the structures are, what they do, and which ones are in all bacteria and which ones are only in some of the bacteria. So let's get started. We're going to start with the outside of the cell. The first of these is the flagella. It's the biggest structure that we notice on the outside of the cell. You may have seen pictures of sperm swimming and they have that big whip-like tail. That's the flagella. However, in eukaryotes, the flagella is a whip, and in prokaryotes, the flagella spins, like in a 360 motion. The flagella is important because what it does is it helps the bacteria move around its environment. The flagella has three main parts: the filament, which is the big tail part, the hook, which is an adapter that helps it meet up with the part that's embedded in the cell, and then the basal body, which consists of a rod and rings. The rings are parts that help actually hook into the cell wall, and they vary based on the type of cell wall the prokaryote has. You can see that in the picture here. In the picture below, you see that there are four types of arrangements of flagellas. You have monotrichous, lophotrichous, amphitrichous, and paratrichous. Make sure you know what the differences are. I'm never going to ask you which one's which, but I want you to be aware of what the four types are. These four types allow bacteria to move in two distinct ways: running and tumbling. I have put up videos of these in Blackboard because it's a little bit easier to see them when they're moving as opposed to just looking at pictures of it. So check out those pictures in Blackboard so you can get a feel for what running and tumbling looks like. There are three more fibers I want to talk to you about. The first of these is Fimbria, and then you can see in the cell here on the bottom left, it's got those darker hair-like structures coming off. Those are Fimbria. They help the bacteria adhere to the surface of its environment. Pili is that dark red fiber that's coming off the cell. This allows for the exchange of genetic information during conjugation, so that plasmids can pass between the two bacteria cells this way. The last structure is actually a eukaryotic structure, but I know you've heard of it before, and so I wanted to address it here, and that's a cilia. They're hair-like structures on the outside of eukaryotic cells. They are not on prokaryotic cells. So remember, the outside structures on prokaryotic cells are pili, Fimbria, and flagellum, and the outside ones on eukaryotes are flagella or cilia. And not all have all of them. Some of them only have one or two. Let's talk about another outside structure, the Glycocalyx. This helps bacteria adhere to their environment as well by forming this kind of sticky substance made up of polysaccharides, carbohydrates, and proteins. And it creates a place for the bacteria to adhere. Have you ever woken up in the morning and had this slick, slimy feeling on the outside of your teeth? That's a Glycocalyx being formed by the microbiome of your mouth. And that's what you brush away. So it helps kind of keep everything there and in place and helps that bacteria adhere. And each bacteria has its own unique chemical composition, so it's not important to know the really distinct specificities about them. Just understand the big overarching purpose of it. Now, when the Glycocalyx is wrapped around the cell specifically, that's known as a capsule. And that forms a mucoidy look on a Petri plate. And you can see that in the picture here on this slide. The bottom two quadrants show you what the mucoid look. It looks kind of like snot on the plate in comparison to cells that haven't developed the capsule yet. The picture here is from the same organism. And so it's just whether or not the capsule has been formed. We can do a capsule stain or a negative stain to see the capsules, and they're just, once again, the Glycocalyx forming closer bands around that cell to protect it from the environment. Alright, so now let's go inside the cell. The first thing we're going to talk about is the cytoplasm or the cytosol. This is the jelly-like substance that is found inside all the cells. It holds all the internal components, so the ribosomes, the DNA, the plasmids, anything like that, as well as protein, salts, nutrients, other things like that. Almost all reactions in the cell occur within the cytoplasm, and it's about 70 to 80% water, so it's pretty jelly-like. So now let's talk about the DNA of the cell. The DNA of bacteria is in a circular chromosome format. You can see that here. Now, it's really big and it'll get tangled up into a jumble, and you'll see that in models where it looks like a pile of spaghetti. But when it's all laid out, it's one big circle, as opposed to us who have 46 linear chromosomes. Bacteria can also create plasmids. These are small little circular DNA that contain specific genes, and these genes can be transferred between cells during conjugation. We also use plasmids in biotechnology, and this is how we inserted the insulin gene into E. coli to create insulin for diabetics. The ribosome is responsible for helping turn the DNA from the chromosome that's been copied into protein to be able to perform the functions of the cell. If you've heard about transcription translation before, you should be pretty familiar with how it works. But there's two basic subunits, and they come together, and they are an enzyme that's made of RNA and proteins to create a protein structure to do the important processes of the cell. We're going to talk about these more when we get to transcription translation in the next unit. Inclusion bodies are a weird little thing, and some cells have a bunch, some cells have none, some cells have only a few. It's very cell to cell specific, not even species, just cell to cell. These are storage bodies, and they're non-membrane bound granules, and they just store nutrients, gas, minerals, anything the cell might need. It's just an extra storage space, kind of like our fat cells in our body. And the last thing we're going to talk about are endospores. Now, it's really important to remember that not all cells make spores, but spores are essentially a way of survival for these cells. They package up their information into a separate little capsule that then they spit out of the cell. And this spore is then resistant to heat, radiation, and even chemicals. They're very hard to kill and even harder to detect. This is why people use spores in bioterrorism. What happens is when a cell detects that the environment is not friendly, it will start creating the spore, and then the spores will be released. And when the environment changes and it shows that it's a good place for stable life, the spore will then germinate, becoming a vegetative cell that will then be able to go on and do the normal life functions of the organism. So remember, these are very hardy little cells that don't actually do the normal characteristics of the species. It's more of just a survival pod until things are ready. One example of this is that spores from Clostridium can infect wounds and cause a release of toxins and can really cause serious damage within the body. And so this is why spores are often the most difficult thing to get rid of in a sterile environment. This is the end of this topic. Please review the objectives and let me know if you have any questions.